It's my honor to introduce Dr. Lane. Dr. Clifford Lane is Clinical Director and Deputy Director for Clinical Research and Special Projects for the National Institute of Allergy and Infectious Diseases at the NIH. In this capacity, he oversees an array of clinical and research activities. Dr. Lane is a fellow Michigander. He's a native of Detroit and received his BS and MD degrees from the University of Michigan, following a residency in internal medicine at the U of M Hospital. He joined the NIH for fellowship training in infectious diseases and immunology and has been there ever since. Dr. Lane's primary research interests are in the areas of HIV/AIDS and emerging infectious diseases. His HIV research activities range from fundamental basic science to large-scale international clinical trials. In the area of emerging infectious diseases, he has been involved in studies of unexplained fever, influenza, Ebola, and COVID-19. Dr. Lane is one of only 2 US scientists to tour 5 Chinese cities in February 2020 to get a firsthand look at the COVID-19 epidemic. He will discuss COVID-19 and what he learned during his trip to China. He also will discuss the clinical manifestations of the disease and advances in treatment, prevention, and vaccines. We really appreciate the fact that Dr. Lane is taking the time to talk with us today. And Dr. Lane, the floor is yours.
Well, great. Well, thank you, Tom. Thank you for that very kind introduction. I'm going to grab the screen. So hopefully that shows up okay.
Looks great.
Good. All right. Thanks. So again, what I'd like to do is sort of cover an array of topics related to COVID-19. I think as most of you are aware, this new disease really, I think, came to our attention in the US early in 2020, when we began to hear reports of cases of pneumonia occurring in China, and particularly in the city of Wuhan in the province of Hubei. Those initial reports suggested that this was linked to the seafood market in that area, although I hasten to add that that epidemiology is a little bit less clear as we obtain more and more information. And I think many people feel now that probably the first cases of this disease really were earlier than December of 2019, maybe even several months earlier. Again, there has been no source identified. I know the origin of the virus is a big topic. I'm not going to go into that to any detail, but I'm happy to take questions on that. I really think what was amazing among a sea of amazing things related to this new disease is that really within a matter of weeks of there being clear recognition that there was a new form of pneumonia in China, in Hubei, that the Chinese scientists had identified the virus that became known as SARS-CoV-2 as the etiologic agent. And they, in fact, were able to sequence that and provide the information needed by others to create diagnostic tests, to work towards therapeutics, and to create vaccines. So things did move really quite quickly in that regard. Just in terms of terminology, we use the term COVID-19 to name the disease that is caused by the virus, the novel coronavirus, SARS-CoV-2. And here you see 2 electron micrographs of the virus on the left, a transmission electron micrograph, so basically a cross section of the virus. And here you can see these little blue spikes around the surface, sort of giving it this image of a crown, hence its name coronavirus. Over on the right, you see a 3D image from a scanning electron micrograph. You can see, this virus really is basically small spheres. They're budding on an infected cell. So the US really became involved somewhere around January 20th, and there were 2 things happened around that time. The first was that the first case was diagnosed in the US in Washington State by a recent traveler to Wuhan, China. And at the same time, the Diamond Princess set sail from Yokohama. And 5 days after that, it was noted that a patient who had gotten off the cruise ship in Hong Kong had been diagnosed with COVID-19. That ship then became quarantined. It gave us one of our first clear epidemiologic looks at the illness. Among the 3,711 passengers and crew, 19% of them became infected. Of note, and something that really wasn't appreciated early on in the outbreak, but clearly was seen here, was that patients, individuals could be infected without symptoms. And while it wasn't documented here, it was documented within a few months that those asymptomatic infected individuals could indeed pass on infection. And of the patients who were infected, 18% of them never had any symptoms whatsoever. And of those who were infected, there was about a 2% mortality, a figure that's pretty close to what we still see today, somewhere between a 1% to 2% mortality. So with this appearance of COVID-19 basically in the US, with there being US passengers on the Diamond Princess, a variety of activities went into high gear, some of which had actually already started. So scientists here and elsewhere used the sequences that had been published by the Chinese scientists to begin work on vaccines, in particular the RNA vaccine. The one we worked on here is the one that was eventually created by Moderna, making monoclonal antibodies to the virus using cells from survivors. The one from here was then commercialized by Lilly, others by Regeneron, Vir, GSK, AstraZeneca. A whole array of companies now are making monoclonal antibodies and vaccines. We began a randomized controlled trial of one of the few drugs that we had available that had evidence of activity against SARS coronavirus 1, and was then demonstrated to also have activity against SARS coronavirus 2, a drug called remdesivir, and began a randomized controlled trial. And as was mentioned, I had the opportunity to be part of the WHO mission to China mid-February 2020. So it was interesting because I was actually headed to Japan. I was at the airport, at the boarding gate at Dulles Airport, getting on a plane to Tokyo to have Japan be a site for a randomized controlled trial of remdesivir, in part prompted by the fact that there were US citizens on the Diamond Princess cruise ship. And as I'm at the gate, I got this email followed by a phone call saying, you know, you've been selected to go on this WHO mission. I said, well, you know, I'm just getting ready to get on a plane to Tokyo. He said, well, you need to report to Beijing in a day. I said, the only way I'm going to be able to do that right now, given that I had no passport to get into China, was to fly to Tokyo, take care of what we needed to do to help the Japanese scientists shown there in the picture on the left, get ready to be part of the trial. And then on the right with my colleague, Dr. Hiromi Imamichi, I go to the Chinese embassy to get a passport to enter China and to be part of the mission. That mission was really quite an interesting experience. It consisted of 25 different experts. You can see us all there on the left in masks. It was, as is typically the case for WHO, a WHO mission. Half of the scientists were from the host country, from China. Half were from outside. In addition to China, there were scientists from Germany, Japan, Korea, Nigeria, Russia, and Singapore. A colleague of mine from CDC was also part of the mission. It was headed by Bruce Alward of WHO, who's a Canadian citizen, and Wanyang Lang of the People's Republic of China. The full report from the mission is published online. I would add that, as is often the case with these type of multinational things, groups, missions, the part to read is in the middle, where you can look at the data and draw your own interpretation. Some of the language that one puts in descriptively may not always be quite as objective as the actual data. So anyone with a strong interest in this, I really encourage you to read that report, because I think it had a lot of very interesting data. some of the facts that mentioned that were reinforced by the visit, that they had their first diagnosed cases from early December 2019, as opposed to what was being published at the time, late December, and I think now even earlier than that. And very clearly, not all of the initial cases were linked to the seafood market, suggesting that there were cases prior to those initial cases from somewhere, even potentially outside the region. And again, what you would typically refer to as case zero or the actual first case still has yet to be identified. As I mentioned, very quickly, they identified the virus, they sequenced the virus, they put the sequences out. In China, they had diagnostic tests developed within 2 weeks. At the time we were there, February 20th, there had been 75,465 confirmed cases throughout the country. Just to put that number in perspective to a number I'll show later, at many days of the outbreak in the US, a single day had more than 75,000 cases. And sadly, that's where we are at the current moment as well. At the peak, one fever clinic in Wuhan was seeing 500 patients per day. And I think many of you may remember that early in January, I can remember this vividly because it was the first time I thought there's something serious potentially serious going on, was that in Wuhan, they built 2 new 1,000-bed hospitals, in a matter of a week or two. Again, from the Chinese side, this clearly was something they were taking very seriously. Now, they did an amazing job in what we would call standard public health measures of social distancing, isolation, and quarantine, not always voluntary. This is just a picture from one of the stops in the mission where we were wearing masks the entire time, except when we were eating. You can see that they had a social distance. While that's set up there in the back, like a buffet, in fact, we were given trays of food and there was very little mixing of things. There was an amazing public awareness of the status of the epidemic within China. We visited a whole array of places, and every place, this was the major topic. How many cases today? How many cases in our neighborhood? This is from one of the living complexes. I can't read what's on the sign, but what I was told was it had the daily case counts for the country as well as for the local region, and in addition for the living community that's represented here. We were on a bus, and again, we were on several buses so that we weren't, all sitting together. And every so often, the bus would be stopped. Any transportation might be stopped. Someone would come on and do a temperature check. I'll add that it's not really clear whether or not temperature checks have a big impact on themselves on limiting the spread of an outbreak, but they certainly cause people to be keenly aware that something is going on and pay attention to it, such that if you had a fever, you probably wouldn't be going out. This is just a look at the outbreak in one of the cities that we visited, Shenzhen. The reason I'd like to show this is it spans a period of December 25th to February 14th, and that's about the time that our mission started. It really is a period of time that is less than 2 months, during which time an outbreak started in this city. They immediately put pretty strict measures in place, including a closing of their schools. And within a very short period of time, this came under control. This is what one might have expected with a SARS-1 outbreak or with the MERS outbreaks that occur, sort of a time limited, maybe several hundred cases, certainly nothing like, what we ended up experiencing with COVID-19. And again, just a demonstration that the usual public health measures can do something pretty effective if they're applied consistently and applied uniformly. This is in the green is the outbreak in China. The big peak there is just a change in the case definition. So there was a sort of a surge of cases that one day, which would have been a shift in the curve. And you can see that outbreak in China coming down as the outbreak in the rest of the world took off. Here you see it in the Americas. What I'm going to show in the next slide is basically I'm going to take this graph and I'm going to paste it into a graph of the global outbreak of COVID-19. So this thing you can barely see down here in the bottom left, that really was the outbreak in China. And then to the right, you see the outbreak throughout the world, the outbreak in the Americas in the top here on the yellow. And again, it's just amazing how we've seen these repeated waves of the outbreak, often related to, in the case of the US, changes in how clustering, going from summer to winter. And I think most people believe this most recent surge that we're really just in the beginning stages of right now are very likely in large part due to the Delta variant. And I'll come back to that a little bit later in the talk. So what I'll do now is sort of break the rest of the lecture into these different categories: epidemiology, virology, transmission, diagnostics, clinical course, therapeutics, and vaccines. These are the numbers from the US as of 2 days ago. Just over 34 million cases, 600,000 deaths. And, when I was putting the talk together, I was certainly hoping that I'd have, almost imperceptible bars here for cases in the US by this point in time. But again, we are really, in the middle of another, or I'm in the beginning, in the beginning of another surge. The deaths haven't shown an uptick yet, but typically the sequence of events is you see an increase in the number of cases, the number of infected individuals, followed by an increase in the hospitalizations, followed by an increase in the deaths. So it's little doubt in my mind, but that we will begin to see an increase in deaths in the not too distant future. This shows the case fatality rate from the March of 2020 up to the present time. What's interesting, and you see this in other outbreaks of other emerging infectious diseases, this would be the same sort of thing that we've seen, for example, in Ebola, that when a new disease hits, particularly if there's not anything quite like it around, it takes a while for the clinicians caring for the patients to figure out how to provide the best standard care. When do you use a ventilator? Are there positions for ventilating, on your stomach or on your back that may be a bit better? What antibiotics do you use? How well do you manage fluids? At what point do you intubate? And again, you can see case fatality rates going from around 6%, this being the number of deaths over the number of cases, and then that dropping down to where it's been, for quite some time now, somewhere around 2%. We have pretty good ways to capture those who are hospitalized and those who die of COVID-19. We don't have a good denominator for total infections. And I think many people feel there may be even twice as many infections as those that we actually capture. So the overall case fatality for this disease is probably somewhere in the range of 1 to 2%. If one looked at this disease at the beginning of 2021, it was the leading cause of daily deaths, comparing it to other leading causes from the prior year. So again, you can see at the peak, really for us in the US, we're seeing somewhere around 3,000 deaths a day. Compare that to around 2,000 deaths a day for heart disease. If you compare the total number of deaths in the US from COVID-19 to the other influenza pandemics in 1918, 1968, and you compare that to the major wars of the US, you can see that we are fast approaching the number of deaths that were seen with the 1918 influenza pandemic, and clearly more deaths than any of the wars that we've been involved in, even when you add them together. So it had a big toll. There is a difference in terms of mortality from COVID-19 based on race or ethnicity. You can see here, looking at number of deaths per 100,000 population, that the Black or African American population, the American Indian, Alaska Native population are disproportionately affected compared to the white or Asian populations, with Hispanic or Latino and Native Hawaiian and Pacific Islander somewhere in between. So again, there is disparities. These are likely more related to disparities in healthcare than anything related to host genetics or genomics. And it's something that we try very hard to remain cognizant of as we look at programs to get out there, whether it's the research side of testing things like therapeutics and vaccines or the implementation side. There are special segments of our society who are particularly hard hit by this outbreak. Moving now to talk a little bit about the virus. I promise we won't go into too much detail, but there are a few things about it that I think are worthy of note. It's the class of virus called a beta coronavirus. It's the same subgenus then as SARS coronavirus. It's the same as certain common cold viruses and some bat coronaviruses. I'll show that in another slide in a moment. It is an RNA virus. It's a virus that buds off the surface of the cell, as you could see from that scanning electron micrograph. It's a positive sense virus, which means you can read the proteins of the virus directly off the RNA. It's a fairly large virus. It has 4 main structural proteins: the spike or the outer protein. This is the protein to which the current vaccines mount an immune response. The envelope protein helps facilitate the virus budding off the cell. And then it has a matrix protein to help hold it together and a nucleoprotein to help hold the RNA in place and package it within the envelope of the virus. To the right is an X-ray crystallographic depiction of the spike protein. This protein binds to the ACE2 molecule on the surface of the cell, angiotensin converting enzyme 2. Then is the receptor for this virus. And you hear that thrown around fairly often in some of the reports about the virus. This is a phylogenetic tree showing where SARS-CoV-2 is here highlighted in the yellow and how it is related to SARS-CoV-1 and MERS. It's also related to 2 viruses that cause the common cold, 2 other cold viruses here. Human coronavirus 229 and human coronavirus NL63 are alpha coronaviruses. So again, there's actually some speculation that these common cold viruses, which go back before we really have good records, actually could have been prior pandemics that the host has just adapted to, either through development of immunity or other adaptations in receptors or responses. There's a lot of talk about bats, and you can see there's the most closely related virus to SARS-CoV-2 is indeed a bat coronavirus. This is what I think, provides a lot of the evidence behind the hypothesis that this really is a virus that jumped, from one species to humans. Most people would think a bat virus went into another mammal and from that other mammal into humans. There are no bat coronaviruses that look like they may have gone directly. Some of the research trying to understand different pieces of bat coronaviruses has given rise to all this discussion some of you may have heard about regarding gain of function. I'm not going to go into that either, but I am happy to take questions on that. Regarding the transmission of this virus, it is predominantly through exposure to respiratory droplets that go from one person to another when those 2 people are relatively close, and sort of the arbitrary 6 feet is what is typically put forward as the distance to maintain between people when one's worried about transmission. Sometimes particles, smaller than particles, aerosols may stay in the air for a bit longer period of time, travel a little bit further. So, there may be some cases, in other words, there isn't like a 100% at 6 feet, there are no more viruses. It's a gradient, and a lot of these public health recommendations are really based on information and a little bit of arbitrariness. Unlike some other viruses, flu in particular, it doesn't appear that there's much spread of this virus through contaminated surfaces, or what are called fomites. It does appear that this really is mostly respiratory, while the virus can be found in a variety of body fluids, including stool, blood, semen, tears, it doesn't appear that that is a significant form of transmission. It is very important to note, and I think this is probably one of the things that has made it so difficult to get this under control, is that a substantial proportion of people who are infected are asymptomatic, but nonetheless capable of transmitting the virus. The amount of virus that people can detect in an asymptomatic person versus a person with disease aren't that different. And in fact, you only really see the virus early in the course of infection, usually before people are really sick. So it's an interesting dichotomy we work in, that you've got a viral replication phase followed by dealing with the damage that's occurred to the host tissue through that early phase of infection, followed by the host immune response to that infection. And people who are asymptomatic at the time they get their test, about 3/4 of them actually will remain asymptomatic. So, you know, it can't be, well, I was exposed, but I feel okay, so I don't need to get tested. I mean, that's exactly the person who probably should get tested or self-isolate, because that's the sort of person who might be unknowingly continuing to spread infection. Overall, and again, these are estimates, it's thought that somewhere around 60% of all infections do result from asymptomatic transmission. About a majority of those from people who will develop symptoms, 35% of that 59%, it made up of 35% of people who haven't developed symptoms yet, and 24% of that 59% are individuals who never develop symptoms. That 59% number. There's a lot of talk about face masks and, you know, what do they do? How good are they? To me, this is just more a common sense thing than any, you know, sophisticated engineering. If two people are close to each other and one has COVID-19 or SARS-CoV-2, that's going to be the highest risk of transmitting it to another person, and a higher risk the closer they are together. If that person who's infected puts on a mask, that really decreases the rate of infection a lot. And this is probably the most important thing that masking can do. If the person who's not infected wears a mask, that cuts it down a little bit. Estimates are by about 1/3, but it's not all that effective compared to the infected person wearing a mask. If both people wear a mask, that's really low. And if both people wear a mask and socially distance, that's basically the lowest we have right now for standard public health measures. And again, I think as you've heard in the last couple of days in the statements from the CDC, while there has been a trend towards liberalizing taking off the mask, I think as Delta is coming in, I think a lot of people, I would say myself included, you know, are being more conscious of putting masks on in more places. Among the ways to prevent acquiring SARS-CoV-2, obviously, right now, I think everywhere in the world, you know, the top 10 things to do really are to become vaccinated. Again, wearing of facial masks, maintaining physical distance, avoiding crowds. If you are going to be gathering, if you can, do it outdoors. We always say frequent washing of hands, but again, most of the transmission appears to be through direct respiratory. We fortunately have a lot of good tools for diagnosing active infection with SARS-CoV-2 or past infection. There really are 3 main types of tests that are used. The molecular tests detect the RNA of the virus. So when you hear people talk about PCR, polymerase chain reaction, these are tests for the RNA of the virus. This is typically felt to be the gold standard. As with most of these tests, the sample is obtained by a nasal swab. That swab then is put in some sort of transport media that goes to the lab where they run the PCR reaction. A little bit less sensitive, but still pretty good, are the antigen tests. So these don't detect the RNA of the virus. They, in fact, detect the ones that are out there now, the spike protein of the virus. Again, it's the same sort of thing. You do the nasal swab, but here you then put it, you know, typically in some sort of cartridge that has within it antibodies to that spike protein. You put some fluid in it, that the fluid will bring the antibody up over where you've put your sample. If the spike protein is there, the antibody gets held up, the antibody has a color with it, and you'll see that color. You actually can buy these tests for home use. I think there's 2 of them that are out there. They're really quite simple to use. And they're really quite convenient. Then finally, there are the antibody tests. So when one gets infected with an agent, a virus, a part of the host immune response is making antibodies to the virus. Those antibodies bind to the virus and prevent it then from infecting new cells and hopefully limiting tissue damage. We can, in the test tube, measure the presence of these antibodies by seeing whether or not they bind to the proteins of the virus that are plastered in different places in different wells or in test tubes. These really only tell you whether or not you've had past infection. They don't tell you anything about whether or not you have active infection, although typically speaking, as you make an antibody response, the virus goes away. And most everyone who has high titers of antibody, by that time, there's no more virus being shed. We can measure antibodies to different parts of the virus. As I mentioned, the vaccines are directed toward the spike protein, but it's not directed toward these internal proteins like the nucleocapsid. So you can do one antibody test for antibodies to the spike, another antibody test for antibodies to nucleocapsid. If both of those are positive, that would indicate past infection. If only antibodies to spike are positive, that would indicate someone who's been vaccinated. Moving on to the clinical course of infection. This is a disease that has many different clinical manifestations. As I mentioned, it goes from patients who have absolutely no symptoms, and probably I should make this particular bar a little bit longer to imply that there are a large number of people infected with no symptoms, to ambulatory patients with mild symptoms, to people who need to be hospitalized requiring low flow oxygen, high flow oxygen, or mechanical ventilation. You can see here in the middle, a CT scan, a slice through the lungs of someone with SARS-CoV-2 pneumonia. You can see all these different infiltrates within the lung tissue. This should all be relatively black, and that white is shown here. As I noted earlier, this early phase of the disease is when you have viral replication. In fact, as I'll mention in a moment, that's when the antiviral therapies that we currently have available are going to be most effective. Once you get into these later stages, it appears that it's the host response to the virus, which neutralize it, but in the process, may also be doing damage to cells that have been infected with the virus above and beyond what the virus itself did. So in these later stages of the illness, we found that immunomodulatory strategies, in particular, the corticosteroid drug dexamethasone, can be quite important in saving lives. And other immunosuppressive agents have been looked at in the setting as well. An inhibitor of interleukin-6 called tocilizumab, as well as a JAK kinase inhibitor called baricitinib. Throughout the course of infection, there also is an increased tendency for blood to clot. And blood clotting has been a key part of some of the more serious complications of COVID-19 in hospitalized patients, such that everyone who's hospitalized at least should be getting prophylactic dose anticoagulation. And in some instances, there are emerging data that patients who are sick but not yet critically ill may benefit, in fact, from therapeutic dose anticoagulation. Those who are critically ill, there's been no evidence for benefit for full dose anticoagulation. This looks at how those people who are sick with COVID-19, how they fare. The majority, 80% of them, have mild to moderate disease, really don't need to be in the hospital. About 20% maybe end up in the hospital. These again are data, early data from China. Of those patients who are hospitalized, 3/4 of them will have severe disease, and about 1/4 of them will have critical disease. And overall, again, at the time these data were put together in 2020, the case fatality rate was about 2%. And these relative proportions have held up through today. While we think of COVID-19 as being something that causes pneumonia, it really can affect many other organ systems as well. It's associated with a lot of inflammation. It has been associated with neurologic disorders, again, respiratory distress, problems with the heart, as I mentioned, increased coagulation of the blood, acute kidney injury, and this very unusual multisystem inflammatory syndrome in children, or MISC, that occurs a little bit later, following infection. It's felt to be related to immune response to the virus, but really can be quite devastating in its acute form. Fortunately, there does not appear to be much in the way of long-term sequelae, but it's really too early in our knowledge to be able to make statements that are too definitive in that regard. The disease prior to the vaccine campaigns really was a disease of those over age 65. And you can look at here the number or rates, of hospitalization from March to January of this year, from March of 2020 to January of 2021, based upon age. Again, the 85 plus really having the highest rates of hospitalization. We're seeing quite a different trend now. A large part of this is because this older population is much more vaccinated. The vaccines are not available on emergency use authorization at this point for children 12 and under. And I think part of that shifting host protection is being reflected in what we're seeing now in hospitalizations. When you couple that with a virus that appears to be more transmissible, you have the picture that we're currently experiencing. Certain pre-existing medical conditions appear to be associated with an increased risk of severe illness with COVID-19, among them cancer, chronic kidney or lung disease, diabetes, Down syndrome, heart conditions, immunocompromised state from immunosuppressive therapy to prevent transplant rejection, obesity, pregnancy, sickle cell disease, and smoking. So when we see an infection in someone with these conditions, we try to jump on that early. Indeed, these are the individuals for whom the monoclonal antibodies are indicated for outpatients who have been diagnosed with SARS-CoV-2. The late sequelae of this disease are a subject of intense study. There's a lot of discussion about this in both the scientific and the lay media. There's brain fog, sort of trouble concentrating, some claims of long-term damage to the heart. I would say that when you have so many people who have been infected with an illness, you're going to have an overlap between some bad diseases not related to that infection and the infection. And what we're trying to do right now is really sort out, what appears to be COVID-19 related and what is not, so that we can target interventions most appropriately. What is clear is in studies that are ongoing, comparing COVID-19 survivors to people who would serve as controls, typically household contacts of those survivors, but people who had not gotten infected, that probably about half of the people who recover from COVID-19 have some sequelae beyond the acute illness. How long that will last, we really don't know at the moment. There isn't any classic case definition that has emerged yet. And part of that, I think, is trying to sort through what really is COVID-19 related and what in fact may be related to other things in the same individual. There has been a very intense effort to try to identify treatments for COVID-19. Thus far, there are two things that I think have been quite successful that I'll mention specifically. One is the use of the anti-SARS-CoV-2 monoclonal antibodies in ambulatory patients who have been recently diagnosed, who are at high risk of disease progression. So overall, those individuals may progress to hospitalization somewhere around 6% of the time if they get one of these monoclonal antibody combinations, or in the case of the Vir antibody, a single antibody, that goes from 6% down to around 2%. So that's a big difference. So that's something that we encourage everyone to be aware of. I mentioned that early on, we started a clinical trial of the drug remdesivir. That study actually showed clinical benefit from the perspective of fewer days in the hospital. It was not large enough to make clear statements about impact on survival. It does not appear to have much benefit to patients who are severely ill, those on ventilators. But in that earlier stage, while there still is some viral replication going on, it looks like it does have some definite benefit there, and it is something that's recommended for use in that particular setting. Convalescent plasma, there are a variety of other antivirals currently in clinical trials, both targeting the polymerase enzyme of the virus as well as the protease enzymes of the virus. Convalescent plasma is available for use under an emergency use authorization. It is a very logical thing to think about. Well, someone recovered, that means their immune system did a good job. I can grab part of their immune system from their plasma, namely their antibodies. Why don't I infuse that into a patient? There isn't that much in the way of antibody in convalescent plasma, and the clinical trials that have looked at convalescent plasma really have not shown benefit. So I think the popularity of this, even though it's available, has fallen way off. And, in contrast, the monoclonal antibodies in outpatients in the randomized controlled trials have shown benefit. Interestingly, these antibodies have been tested in hospitalized patients and shown to not be of benefit. However, thing that really, information over the last days to weeks, if you separate the hospitalized patients into groups based upon whether or not they have already made their own antibodies, you find there probably is a subset of hospitalized patients who will benefit from these monoclonal antibodies, namely the ones who have not yet made antibodies on their own. So that second phase of the illness, when the host has responded to the virus, when there's tissue damage, perhaps related to some of this host response, the drug dexamethasone has been shown to improve survival. And this is strongly recommended for patients who are hospitalized with COVID-19 who have an oxygen requirement. It's 6 milligrams of dexamethasone daily for 10 days. In addition, for some patients who are showing rapid progression during the time of rapid progression with evidence of markers of inflammation, the drug tocilizumab is recommended. Similar comments for baricitinib. Other immunomodulators are under study. And as I mentioned, anticoagulants are in trials. Everyone hospitalized probably should at least be on prophylactic doses of anticoagulants. Clinical trials are suggesting full dose anticoagulation, not of benefit, potentially of harm in severely ill patients, may be a benefit in patients at earlier stages of severe illness. This has been an area that has been really difficult to sort out. And if you follow COVID-19 in the news, you'll see every few days some claim of some new drug being of benefit. There are over 2,000 studies listed in a website called clinicaltrials.gov that lists all the ongoing clinical trials, at least that are either funded by the federal government or regulated by the federal government. It's a requirement that they be listed there. In an effort to try to bring this information in a more digestible form, there are a set of COVID-19 treatment guidelines that are sponsored by the NIH. They are updated very frequently. This is just a summary of some of the, give you an idea what those current recommendations look like. Again, for the hospitalized patients, dexamethasone. For the less severe patients who are hospitalized and require oxygen, remdesivir. Hospitalized not requiring supplemental oxygen, the recommendation is against using dexamethasone, because in the clinical trials, that group actually was found to do worse. So there's quite a bit of nuance in deciding what to use for treatment. These guidelines have been really quite popular. They were first released in March of 2020. We've had 25 major revisions since then and over 15 million page views. So I'd like on the last part of the talk to speak a little bit about vaccines. This is something that certainly has been in the news in the last few days quite a bit. This really is, I have to say, in my career in biomedical research at NIH, one of the most astounding accomplishments that I have ever seen. The fact that we were able to get a vaccine deployed under emergency use authorization in under a year really is an amazing feat. This was indicated by Science Magazine as the scientific breakthrough of the year for 2020. You're likely aware there are really 6 vaccines that are the main focus of study and deployment in the US. They employ 3 different platforms, the nucleic acid or the RNA vaccines, adenovirus vectors, in other words, taking a virus, crippling it so it doesn't replicate, and then putting in a new piece. In this case, the spike protein, the nucleic acid vaccines have in the RNA, piece of RNA that encodes for the spike protein. And then the final approach to vaccination is sort of the old school. Well, real old school is killed virus. Old school is recombinant proteins of the virus. So again, the recombinant protein and adjuvant. In terms of speed of development, the nucleic acid ended up being the quickest to develop, followed by the recombinant viruses, followed by the recombinant proteins with an adjuvant. The adjuvant enhances the immune response. So the two companies with the RNA vaccines out there are Moderna and Pfizer BioNTech. Both of them showed incredibly high efficacy versus symptomatic disease. In other words, any symptom, not just hospitalization, not just severe disease, any symptom at all of COVID-19, 94, 95%. This is in the very highest level of efficacy we see of vaccines. This is what we see with measles. This is better than what we see with most anything else. I mean, if we had an influenza vaccine that was this good, and I can guarantee you these companies working in the RNA platforms are looking to see what they may do in other infections like influenza, we would not have the impact of influenza that we do each season. So these really look quite good. The adenovirus vectors, while you can't really compare the studies of the adenovirus vectors to the studies of the RNA vaccines because they were done at different points in time. So you had the RNA vaccines coming out first before there was much in the way of variants. The adenoviruses came out once the alpha variant really was taking quite hold in some of the sites where the study was being done. And you see for the adenovirus vectors, that efficacy overall is somewhere more in the range of 80%. The two RNA viruses and the J&J Janssen vaccine, I'm sorry, the two RNA vaccines and the Johnson & Johnson vaccine are, all three of these are available now in the US under emergency use authorization. AstraZeneca has reported their phase 3 results. They've indicated, I think I even heard something today that they are planning to file for emergency use authorization in the US. A bit behind all of those then are the protein vaccines. Novavax has reported out 90% efficacy versus symptomatic disease. So again, that's great. And the GSK Sanofi vaccine will expect some results likely by the end of the year. And depending on those results, you know, see whether or not it goes to emergency use authorization. We'll talk a little bit in a moment about, you know, the potential role of booster vaccination and mixing and matching these. What I can say is we don't have a lot of information. Scientifically, there really isn't any real reason to think it would be a problem. In fact, there are some reasons to think that if you used a different backbone for your boost from what you did from your primary, you may direct that booster response much more to the relevant protein, namely the spike protein, rather than anything else that might be there within the backbone, particularly in the case of the viral vectors. As I mentioned, this was like an amazing sequence of events from January 10th, when the virus sequence was released, to having candidate products. This is for the Moderna RNA vaccine that the NIAID Vaccine Research Center played a major role in. In fact, it was basic science on spike proteins of other viruses that allowed the group working on this to figure out the best way to configure that spike protein for the RNA viruses. In other words, it wasn't exactly the way it is in nature. It's modified a little bit to give it a bit more stable of a structure to give you then a better immune response to it. But preclinical evaluation, production of what was going to be the clinical lot occurred very rapidly after that. Phase 1 studies beginning about 6 weeks after the sequence. Phase 2 a few months later. Phase 3 a few months after that. Initial results about 4 months after that, and the EUA submission on November the 30th of last year. So this really was record time. And there are a lot of discussions now about, okay, if we end up in this situation again, you know, can we shorten this timeline even further? These are the data from the first 2 vaccines to complete phase 3, the Pfizer RNA and the Moderna RNA. What is amazing to me is how similar the data from these 2 trials are. Again, both of these are 2-shot vaccines. The Pfizer vaccine has a boost of 21 days. The Moderna vaccine, at 28 days. They are very, very similar responses. So the attack rate, the incidence of COVID-19, the cumulative incidence of COVID-19 is shown by the red lines. You can see the numbers just going up in the placebo group here in the red line compared to the vaccine group. Once you were about 10 days out, even from that first dose, you had some degree of protection. Similarly, for the Moderna vaccine, once you were about 10 to 12 days out from that first dose, you had evidence of protection. These are like amazingly effective vaccines. The main question today is how long does this protection last? And how good is this protection against circulating variants, in particular the Delta variant? We're all hoping that we will get to a point where this magical entity of herd immunity occurs. It's not entirely clear when or if that will be the case, but it is something certainly to be aware of. It's certainly defined as the fact that if enough people in a population are immune to a certain infection, that basically that infection will be unable to spread. There will be no more susceptible hosts. Now, the thing that I don't think often gets discussed enough is there are a lot of variables that play into herd immunity. So people say, well, we need 70%, or we need 80%, or we need 85%. It really depends on how infectious the virus is and how good the immunity is. So yes, we strive for 70% vaccination, but who knows what that number is or if there even is a number. So I just think it's important to put that perspective on it. Again, challenges to getting to a point where we actually don't have this, in essence, as a daily threat is the vaccine hesitancy, people being unwilling to get vaccine for a variety of reasons, some of which are actually understandable, mostly I think due to not having enough good information out there. And by good information, I mean accurate information from trusted sources, and different populations of individuals will have different trusted sources. We don't know how well the vaccines will prevent transmission. Again, these are systemic vaccines. They're really good about preventing someone from getting sick, but what they do to replication at the mucosal surface, let's say in the upper respiratory tract, it's hard to say. These vaccines make good IgM and IgG antibodies. What you need are IgA antibodies to protect the mucosal surface, and we don't have much data yet on how good they are at that. People have a variety of anecdotes out there about viral loads in vaccinated versus unvaccinated. I think it's really not clear. So I think we don't know what role vaccines will have. Certainly, they will minimize the number of people who get sick. That certainly is true. We have the challenge of the new variants. And then now, with the initial EUAs back in December, we're, 7-8 months out now, and how long we will have protection from that first, series of vaccinations, and whether or not we need a boost, that's something that is a very active daily topic of discussion, trying to get as much information as we can at that. The variants are really much more of a challenge than I think anyone thought that they would be. Across the board, viruses are pretty simple life forms. They're strands of nucleic acids, either DNA and RNA. They can't replicate on their own. They need to get into a cell and steal some of the machinery of the cell to replicate. And they realize for them to survive, they have to be able to change over time. All viruses constantly have the ability to change through mutation. The enzymes that orchestrate productions of new copies of the nucleic acid are not perfect. So viruses constantly have this ability to change. For some of them, like HIV, that's something that's been well established, well recognized. If you treat someone with, many of the earlier drugs for HIV, you treat them with a single drug, within 10 days to 2 weeks, that virus will have mutated away and that drug will no longer be effective. For coronaviruses, it wasn't felt to be such a big deal because the coronavirus outbreaks that we've dealt with have been pretty short-lived. When one has studied other coronaviruses, you don't see much in the way of mutation. The problem that I think we ran into wasn't that the prior data were wrong. The prior data never, the prior data were not generated in a setting where you had, basically hundreds of millions of infected individuals. So with a massive number of people who've been infected, there's all these opportunities for changes to take place, in the nucleic acids of the virus. And if a change takes place that lets that virus replicate better, that change is going to stick. And over time, it's going to be the major population of the subpopulations within a community. So the more time that virus replicates, which is related to the more people who are infected, the greater your chances of seeing something that could be a significant variant. Again, just bringing it back to the phylogenetic tree, just on SARS-CoV-2, all these variants are still all SARS-CoV-2. They're just different slight modifications of that particular backbone virus. You can almost think of it, as a person, wearing different clothes maybe, because a lot of the critical variations are indeed in the spike protein. The variations we worry about getting around a vaccine are indeed in the spike protein. So these variants that are coming up have been classified into 3 groups: a variant of interest, a variant of concern, and a variant of high consequence. So a variant of interest is a mutation that has been identified that is potentially associated with increased transmission or decreased severity of disease. Decreased efficacy of diagnostics, decreased efficacy of therapeutics, or decreased efficacy of vaccines, as I said earlier. Thus far, there have been 6 variants of interest identified and followed carefully. Variants of concern are variants of interest that have gone on and demonstrated evidence of increased transmission or disease severity or decreased efficacy of diagnostics, therapeutics, or vaccines. There are 4 of those as of yesterday. A variant of high consequence, is a virus with mutations with clear evidence that prevention measures or medical countermeasures have been significantly reduced in effectiveness relative to previously circulating variants. So fortunately, there are none of those at the moment. The 4 SARS-CoV-2 variants of concern are Alpha, first noted in the UK, Beta, first noted in South Africa, Gamma, first noted in Brazil, and Delta, which was first noted in India. As I think you're all aware that this SARS-CoV-2 Delta variant is currently the predominant variant in the US. It does appear to be associated with increased risk of transmission compared to ancestral strains and the Alpha variant. It is associated with a modest decrease in neutralization by sera from previously infected or vaccinated individuals. But one thing I didn't stress that I'll mention now is we can measure antibodies to the virus pretty easily because we can get that from a blood sample. Antibodies are only one part of the host immune response to a virus, and other elements of the immune system, in particular the T cell limb of the immune response, often referred to as cytotoxic or killer T cell part of the immune response, also plays some role in host protection. So we don't know whether or not measuring the antibodies give us the full picture. What gives us the full picture is, do we see breakthroughs? Do we see people who are vaccinated getting infected? And if so, are they getting preferentially infected with one variant versus another, correcting for the prevalence in the population? If you look at what's happened in the US from April to July, you see we went from the Alpha variant being the main circulating variant to Delta being by far and away the main circulating variant. The data for July 3rd, those are actual data. The July 17th are estimates based on a model that has been pretty good over time. So again, the major circulating variant is Delta. Now, one thing to keep in mind, since the major circulating variant is Delta, if a vaccinated person gets infected, it will likely be with Delta. That doesn't mean that Delta is less protected than Alpha, Beta, Gamma, or any of the others. So you just have to keep that in mind when you hear media reports about some of these things. We try to get a sense of how well the vaccines are working, not just in the clinical trials, but by studying what's going on, in quotes, in the real world, so-called real world studies. And again, here looking at the Pfizer vaccine, studies in Scotland and 2 in England, looking at either infection without some infection overall, infection with symptoms or infection with hospitalizations, you see that the most important thing really is how well we're protecting against severe disease. And in these real world studies, which are now several months old in terms of the data, they are really very effective in preventing hospitalization. Again, the perfect vaccine prevents infection. There are very few that do that. What we need is a vaccine that prevents severe disease, and we certainly do seen that we have that. Symptomatic infection, then somewhere in between. These are pretty much the numbers we would have expected from the clinical trials. Unfortunately, as you know, rates of infection have plateaued in the US. I actually pulled the data from Michigan over here as well. You can see there was pretty good uptake from January through to April, and then from April on, it's been pretty much a plateau. You know, overall in the US, about 58% of the population has received at least one dose of a vaccine, 50% fully vaccinated. The numbers, you know, about the same in Michigan. 58% have gotten at least one dose, and around 54% fully vaccinated. I did notice, looking at the map of Michigan, that within Michigan, it appears that the Traverse Bay area has done really the best job in terms of vaccination. So any of you thinking about local holidays, that may be a place to keep in mind. Breakthrough infections is really the main topic. I think I certainly see that's what's on the media these days. These are infections occurring in someone who is, in quotes, fully vaccinated. In other words, got 2 shots of an RNA vaccine or 1 shot of J&J. You know, these may be due to a variety of factors, you know, something related to the host, waning immunity, the variants of concern. This has led to intense discussions about whether or not there is a need for additional or booster doses. And while there's no recommendations coming out, I think it's very, very likely that within the very near future, there will be recommendations for boosters in certain circumstances, likely for those individuals who are immunocompromised, for whom it appears they don't make as good a response to the vaccines as those who are not immunocompromised. So in summary then, COVID-19 is a new disease, likely originated in 2019. While we have learned much in the 18, 19 months that we've been looking at this, there is still much to learn. We do know it's caused by the novel beta coronavirus SARS-CoV-2. While a majority of therapies exist, mortality remains in the range of 1% to 2%. Several highly effective vaccines were developed in record time. The emergence of variants has led to more infectious forms of the virus, and global vaccination indeed may be the very best path to controlling this pandemic. So thanks very much for your attention. I'll stop the share and be happy to take any questions.
Thanks, Dr. Lane. I do have 2 questions in the chat. If I or a family member is diagnosed, what can I do or what should I ask to determine the medical professionals are using the best and most up-to-date treatments appropriate for me?
So I think the way that the healthcare providers can get the most up-to-date recommendations for treatment are go to that NIH COVID-19 treatment guidelines. That's a frequently updated website. It has some summary tables. I think we're trying very hard as new things are learned, as new data come out from studies, even before they're published in the peer-reviewed literature, we're trying to get guidance out there. And we do have summary tables so I've been diagnosed and I'm an outpatient, I'm an ambulatory patient, you know, what should I get? I'm in the hospital, you know, what should I get? So that I think is the best source that I can provide at the moment.
Okay, thank you for that. When is the FDA likely to give full approval of the vaccines for COVID?
That's a really good question. I don't know is the only honest answer I can make. There's a lot of pressure because I think you're likely aware, if they have full licensure, any physician, NP, independent licensed practitioner can write a prescription for it. It doesn't have to be according to what's on the label. You have that discretion. And a lot of the meds that we use actually are off-label prescriptions. It's a little bit of a catch-22. The group that sets basically US guidance on vaccinations is something called the Advisory Council on Immunization Practices. And that's an advisory committee to the Centers for Disease Control. That group is likely reluctant to recommend a booster if you can't get a booster. And today you really can't get a booster under the emergency use authorization, which is the regulatory framework under which the vaccines are being provided. Having said that, you know, the vaccines are out there, the push is out there, you know, I would not be telling you, you know, what I know if I didn't say, I know there are quite a few examples of individuals who have on their own, you know, figured out a way to get an additional shot. That was long-winded getting back. That was just me avoiding the real question of when does FDA, you know, probably come up with full licensure. And my colleagues at FDA will kill me no matter what I say on this. I would say that we certainly hope that by the time we get to next year, that is something that we're not asking anymore. Let me just leave it at that.
Thank you. We have a couple more in the chat. How much more infectious is the Delta variant, and is there an increased risk of hospitalization and death as well?
Mm-hmm. So it's a really good question. It's not clear exactly how much more infectious it is, because it's a matter of how you measure infection. So if I just do a nasal swab and measure how much virus, and I do it for someone with Delta and someone, you know, with say Alpha or Beta, you know, I will get two numbers, and there are published data that say, well, it's 1,000-fold higher. The problem is if it's 1,000-fold higher on one day and then drops quickly, that may not be more infectious than someone that is at a, you know, 1,000-fold lower, but there for 5 days. So there aren't a lot of good data. The best data is, well, how many people does someone who's infected with Delta end up infecting compared to someone who's infected, say, with Alpha? And those data aren't so clear because you can't do a controlled randomized experiment. So you're dealing with populations. You're dealing now with populations that are variably vaccinated, variably have prior infection. I think most people are guessing that it may be somewhere around 2 or 3 times more by that parameter. And that really is the parameter that's important. How many more people would you infect? Thus far, there are no data to suggest that it leads to more severe illness. So that is obviously, I think, a good thing. And again, as I said, the vaccines do appear to generate antibodies that can neutralize Delta. The higher the total level of antibody, obviously, the better it's going to neutralize Delta. And that is one of the arguments for moving to an additional shot for those of the RNA or the J&J, for those who've gotten the primary series.
Okay, thank you. We have had several more come in, so we'll try to get through. How best to encourage unvaccinated friends or family to get vaccinated?
That's such a good question. You know, I heard something on CNN this morning of a couple of people who were sort of asking that question, who had relatives either who had become quite ill or had died despite pleading. Really, I think it was, you know, one was an aunt, one was a mother, and it's really hard because it depends really on the audience. You know, the audience of don't do it for yourself, do it for everybody else. You know, don't do it for yourself, do it for your children, your grandchildren, your spouse. For some audiences that might help. What we really need is we need to figure out who the populations are who are reluctant to get vaccinated, and figure out who they trust and listen to, and try to get those people getting the messages out. I mean, you've got the one group, I don't have time, or I'm invulnerable, sort of that younger group. I think they may be compelled a little bit more by, you know, helping their parents or grandparents. There's the other group that views this as a political statement, and we need the people that they follow politically, I think, to come out and provide guidance on that, or people who share similar political views. It's really hard, and it's just so sad for me to see, you know, how what should be very separate from politics has become so political.
Thank you. Another question, once you recover from a breakthrough infection, what level of antibodies do you have? Can you get a second breakthrough and transmit the virus?
So another really good question. I don't know any data on the question, but I certainly can extrapolate from other infections that if you're vaccinated and you get infected, you get a big boost in your immune response to that virus. So indeed, probably after a breakthrough infection, you would have an even better immune response, certainly than you did before you got infected. Again, that will change over time. Depending upon yourself, the host, the degree of protection will vary. So again, someone could get infected again. So there's nothing yet as I'm invulnerable because my antibody level is above X. We don't have anything where I can make that type of statement. So again, I think right now, particularly as we're dealing with a Delta variant and increased risks of transmission, I think everyone has to view that they're potentially at risk of being infected. Now, fortunately, everyone I don't think has to worry they're at risk of being severely ill. I think there is differential there. But in terms of being infected and passing infection, I think basically everyone should feel that they carry that risk.
Thank you. What can you tell us about side effects noted post-vaccination, particularly following the second dose in hormonally producing women?
Mm-hmm. So the one thing that has shown up as a very, very rare side effect has been this blood clotting that has been analogous to something that is seen again in a very small number of people who get the drug heparin, called heparin-induced immune thrombotic thrombocytopenic clotting, and it's caused some big clots in like major veins in the brain. It's a really scary thing. There is no question that it's something you would not want to get. It appears to be a bit more common in women of childbearing potential, but it is extraordinarily rare. It is so unbelievably rare that people need to be aware of it. I mean, there isn't something like this that's been seen with the RNA vaccines. It's probably, it's almost certainly real, but it is really, really rare.
Thank you. How long does vaccine protection last? And is the protection as good from the Delta variant as from original COVID?
Yeah, the protection to Delta is probably a little bit less compared to the ancestral strain, but at least at this point in time, it doesn't look to be critically less. In other words, you don't need to have excessive worry about that. The duration of immunity based on your ability, your protection from getting infected, we don't really have an answer to that. We can look at declines in antibody and we can see that the antibody levels do decline over time, but we don't know what the protective threshold is and actually whether or not antibody by itself is the real marker that we should be measuring.
Thank you. Someone comments, excellent presentation. Their one question is, some vaccine doubters believe that shortcuts were taken in developing the vaccine so quickly. How can we reassure that no shortcuts were taken?
I think that the way to think about this, it's a really good question because, right, if I sit here and I brag about the fact that it was done in record time, you know, the flip side of that is, well, what steps did you skip? What I can tell you is there was a massive, massive investment of resources to all of these companies, either for advanced purchase or to support the manufacturing, to support the clinical trial. This to me was one of the great achievements of the Trump administration. They did this Operation Warp Speed. And I'll tell you, they did it like I've never seen it done before. There were tens of billions of dollars that were put into this effort. Large numbers of people were hired. People were hired away from the vaccine companies not doing the work to the vaccine companies doing the work. Basically, staff, my own staff, myself, I mean, people who were working on other things pivoted basically 100% to work on this. So what you had was you have a massive expansion of the workforce, a massive expansion of the funding. And what you did was you did all the things. You just did them really quickly. Some of the things were done in parallel. So, and at risk. So in other words, you would typically, you know, figure out your vaccine. What is it going to be? It's going to be an RNA. It's going to be a protein. Okay. Well, what does this do in tissue culture? Okay, well, it looks pretty good in neutralizing virus. Okay, I'm going to study it in mice now. Well, it looks pretty good. I'm going to study it in larger animals now, you know, guinea pigs, hamsters, non-human primates. Well, that looks pretty good. So now I'll go into phase 1 in humans. Basically, that work was all taking place simultaneously. So in other words, everything was being done really at risk. Well, once I got my phase 3 study, then I'll start ramping up production so I can sell it. What the government did from the beginning was they said, listen, you get a good vaccine, we guarantee you this much purchase. So advanced purchase agreement. So they knew that there would be a purchase so they could start production beforehand. So in other words, the steps were all there. Things that were typically done in a linear sequence, many of them were done simultaneously, and that was able to be done because of really, it was a massive amount of resource that was put into it. And again, put into it by the Trump administration.
Thank you. It seems unlikely to me that so few cases have been reported among the Chinese population. China has some very densely populated areas, including the city of Wuhan. Can we trust the data reported by the Chinese agencies in this regard?
That's a really good question. I think you can apply that to basically any country in the world as well, because no one wants to think they have a bunch of COVID-19. What I will say is that having observed that in China, if you had COVID-19, if you weren't in the hospital, you were in a forcible quarantine in a state-run facility. And if you were a close contact of someone who had a case, you were forcibly quarantined in another state-run facility. This was not an option. There wasn't any voluntary isolation. This was all monitored in a very regimented way. You could see they're building these 2,000 bed hospitals thinking a big outbreak. That just hasn't been the case. You know, where the numbers might be fudged a bit, who knows for anybody. I mean, the numbers I showed you, I pulled from the WHO site. I can pull some from the Hopkins site. I can pull some from the CDC. I'll get different numbers depending on where I go and what the sources are. I just don't think there's a massive outbreak in China, at least in the urban areas, that we don't know about. But, you know, it's, how do you know, right? I mean, it's a fair question.
Okay. Do you think people who were vaccinated early on in January, February may need a booster sooner than a person recently vaccinated who had COVID and was hospitalized? Is there a test for level of immunity?
So the antibody tests will measure your level of antibody. Right now, we don't know what level of antibody might correlate with protection from infection. But I'm guessing, I mean, that may be 6 to 8 months off possibly. So there isn't any test you can use. I think generalizing, your immune response will peak about 2 weeks after your immunization, and from there on it will begin to decline unless you're exposed to the antigen again, either through infection or another vaccination. Some individuals, though, won't make much of an antibody response no matter what happens. Interestingly, when you look at the other beta coronaviruses, SARS, MERS, some of the common cold viruses, not everyone even makes an antibody response to that. More people make antibody responses to these vaccines than make to some of these other coronaviruses. So again, any given individual may be a little bit different from the generalization to the full population.
You clearly indicated there is no proof as to the origin of the virus. So how do we combat the terrible rumors that are being spread?
I think to me, the easiest way to try to deal with that, and you're right, if it was a rumor, if it was a hypothesis without the emotion, it would be so much better. But it all gets tied to an emotion or something being hidden or something was funded that shouldn't have been. When those are just so off base. Yes, since we don't know where the virus came from, anything legitimate should be entertained and should be still on the list of possibilities. I think what you can do is you can look to historical precedent and say, you know what? Other times when this type of virus has entered the human population, in particular, most recently, SARS and MERS, it's been through an intermediary animal host. And by the way, it has taken months and months and months after the first cases, in some instances, years, to figure out what that host is. So I think that, to me, is the best way. It's one of those things, you know, sort of a medicine thing people say when they're doing a differential diagnosis. When you hear hoofbeats, look for horses, not zebras. That doesn't mean there aren't zebras, but it means, you know, the odds are this was a jump from some animal to humans.
Thank you. There are some individuals who have residual effects long after having been diagnosed with COVID. Is it likely that these symptoms will diminish over time?
It's a very good question that we don't know, and again, it's hard to generalize because there will often be exceptions to any generalization. We do find, and again, that's why we're following the cohorts long-term, so we can provide some better information on that. One would hope, and is often the case, that things will get better with time following a viral infection. One of the more recent things we have a cohort of Ebola survivors that we follow in West Africa and Liberia. And those symptoms, while some of them can be quite persistent, and then these were quite ill people, they do seem to decline over time. So I am hopeful that there will be diminution of symptoms in the vast majority of people over time.
Okay, looking back, what do you think the US has done well in responding to COVID? And what do you think we haven't done well?
So what we've done well, I'll sort of put through the prism of the things that I know the most about, namely the research response. And I can go both well and not so well. In the research response, I showed one slide where I said there were 2,000 clinical trials currently listed studying different therapies for COVID-19. When we were in China, I didn't mention this, when we were in China, one of the things we observed was that there were hundreds of clinical trials, but there was no coordinated effort. If an investigator wanted to test something, they just tested it. And there wasn't, and even with direct questioning, well, how do you set priorities? I mean, certainly you don't think 100 of these things are equally worth studying. Well, we let the investigators do what they want. We're busy saving lives. If you don't develop new therapies, you've really missed your big chance to save lives. So in the US, the same thing was happening February, March of 2020. Investigators throughout the country were like doing a variety of different studies, trying different things. We tried, centrally from Health and Human Services, from NIH, to orchestrate and organize some of the clinical research efforts such that the most promising things did get studied first in the largest studies, so that we could come up with some answers, helping us get to better knowledge on the antibodies, on some of the immunomodulatory agents. So I think we did a pretty good job of that. So that was sort of good and bad, that we allowed a lot of things to happen, but we did try to orchestrate it. Stepping out of the research arena, I think the lack of a consistent, clear public health message, with the right caveats, such that when things change, we expected them to change. I think that was our biggest challenge, the thing I wish we had done better. That, it's great to have, news conferences and, provide new information, that if there isn't sort of a collective commitment, in China, this was a war on COVID-19. There was no ambiguity about it. The single most important thing to that population, everyone we talked to, and again, this would be all over the place, and it would be sidebars with people who spoke English, was, following those numbers, doing what they needed to do, getting the counts down. I don't think we ever had that type of consolidated, coordinated, single vision, single mission approach in the US.
Thank you. We do have about 4 more questions and comments. We have about 6 minutes left, and appreciate you just this rapid fire you're under right now. Can you address viral load was the question? Not sure specifically what.
Yeah, so viral load, I do that in 2 ways. So viral load is basically the number of viruses per whatever. So when you do a swab, you basically get virus out, it sticks to the swab, you put it in transport media, and then you run a PCR and you look to see basically how much virus is in the fluid that the swab went into. Generally speaking, the higher the viral load, the more severe the disease. In other settings, there's been no correlation of that for the nasopharyngeal swab and the RNA levels. In contrast, we can also look for virus in blood with PCR or with direct measurement of antigen. In those settings, it appears people with the higher viral loads or the higher antigen, in particular, higher viral load PCR counts or copies of RNA, the higher the viral load by RNA or the higher the viral load by viral protein, in the setting of low antibodies, the more severe the disease. So there is a correlation in the blood between the amount of virus, the viral load, the amount of antibody, and the outcome. High viral load, low antibody, bad outcome.
Thank you. How are other countries doing in getting vaccinated, getting their populations vaccinated?
You know, it's so interesting to see, you know, the US plateauing where it is, you know, somewhere around this 60% or so number, and looking at other countries who are just so desperate to get vaccine. And when they get the vaccine, it's rapidly used. You know, I saw today that right now, the population of Europe on a percentage basis is now more vaccinated than the US. The UK, I don't know the numbers exactly, but I think it's probably quite higher. The low and middle income countries, it's really low. Australia, it's low. Japan, it's low. I mean, these are countries that felt they weren't going to need it necessarily as quickly as other countries. So it took them a little bit longer probably to get their purchase agreements in. So again, we're not the lowest, but you know, we're plateaued, and the way things are going, I can just see many other countries, you know, in sequence eclipse us.
Mhm. Okay, this person says they were surprised that the Delta variant was categorized in the first level of variants. Is it only a matter of time until the most risky variant appears?
That's everybody's worry, right? Everyone's worry is, you know, for lack of a better term, Delta plus. Again, there isn't much evidence that it causes more severe disease, and our countermeasures are still pretty active against it, vaccines and the monoclonals. The worry is if that variant, you know, picks up something else, it could turn into something worse. And that's really, I mean, that's why it's so important to get people vaccinated. It's so important that we do everything we can to diminish spread, and so important for us to get vaccine around the world, right? Delta came out of India. Until the world has got this under control, everyone in the world will be at risk.
Do you recommend one going ahead and getting another shot, which I'm assuming they mean a booster if available?
So I can't really recommend that because it's not part of the emergency use authorization. But I think there's a lot of logic to that. And as with everything that gets into this gray zone, it's best to talk to your doctor about that.
Mhm. And somebody is asking if a copy of the syllabus as well as the recording can be provided. There will be a recording available at the end of the semester for everyone if you'd like to rewatch it. And I won't put you on the spot. We can talk offline, doctor, to see if providing the syllabus would be appropriate. But I had multiple comments throughout the chat of just what a terrific presentation and breadth of knowledge, and the ease in which you communicate and thoroughness is appreciated. So I will I will first ask you, Dr. Lane, if you'd like to open it up for a few more minutes or turn it back over to Tom to close the class.
I have something I need to do at 2:30, so probably I need to turn it over to
Okay.
Tom.
Yeah.
Very
Yeah.
good.
Thank you very much, Dr. Lane. Really appreciate your presentation today, and we admire all the work you and Dr. Fauci are doing.
Thanks. I should add that, right? It doesn't matter how anyone introduces me. The only thing I'm known for around here is that I work with Dr. Fauci.
Yes, your assistant told me that. Yes. Thanks again.
Yes, thank you so much. We appreciate your time and just appreciate the information today. So you
Great.
have a wonderful day. It is now
All right, thank
2:30.
you.
Everybody have a wonderful afternoon.
Yep. Thanks.